I am here for the first time at the Scientists Network, and today I'm going to present a topic which has occupied me for a long time and still occupies me as I work for Wout and Wissen here in the Black Forest. Well, now how did I come to this topic? Actually, it was quite simple. During my studying time, I synthesized a lot of organic molecules, and neither me nor other colleagues had a doubt that making such compounds is tough. Later on during my PhD and doctorate, during my doctorate and my postdoc, I synthesized nucleic acids and proteins. And of course, I was also confronted with the theory of chemical evolution. And it simply boggled my imagination because it totally was in opposite to my experience. I couldn't imagine that nucleic acids or proteins might have been formed somehow by fortuitous chemical reactions, because it was a tough job synthesizing them in lab. So, well, now what is actually the challenge? The challenge is to go from small molecules such as phosphate, urea, hydrogen sulfide, formaldehyde, and so on, and first to arrive at life's building blocks such as nucleic acids, proteins, sugars, and fats by chemical reactions triggered by lightnings and other energy sources such as UV light. And later on, the building blocks somehow are supposed to self-organize themselves to a cell. And well, I asked critical questions to my colleagues, to other students, and also to professors. And somehow I have never received reasonable answers. Some of them even got angry when discussing with me. So today I am grateful to have the opportunity to discuss on some arguments and questions about this topic, which might be helpful for us to have an honest discussion on this theory. Let's start with the building blocks, or say even at a smaller dimension, the amino acids. Amino acids constitute the proteins, and proteins are nearly everywhere in biology. They are an important building material of all organisms, and they are also catalysts that drive the metabolism. And nearly in all organisms, proteins are composed of 20 canonical amino acids. Of course, there are some exceptions, but on the whole, it can be stated that this set is universal in nature. So, and of course, this leads us to the first question: how was the set of amino acids established in nature when we start from evolution theory? And in evolution theory, there are two explanations how amino acids might have occurred on Earth. They may be of extraterrestrial origin or of terrestrial origin. The first ones then are called exogenous amino acids, and the others endogenous amino acids. And usually it is supposed that the canonical set of amino acids is a mixture of both. And of course, in both cases, amino acids are formed by fortuitous chemical reactions. And a logical expectation would be that such a set of amino acids would hardly be optimal with respect to its biological functions. One would rather expect it to be suboptimal. So, and therefore, exactly this question was posed by a scientific group during the last years. The canonical set was compared to random sets which were obtained from a large pool of 1,913 amino acids. This was a purely theoretical work. This means there was no experiment performed under the fume hood, but nevertheless, it is quite interesting because they calculated the coverage of chemistry space of each of these subsets and compared it to the canonical set. In total, they analyzed 100 millions of sets. And out of these 100 millions of sets, 6 proved to be better in coverage of chemistry space. Coverage of chemistry space means that parameters such as size, charge, and hydrophobicity are compared. And the coverage of chemistry space is ideally to be not only broad, it has to be also even. So these are the parameters where the canonical set and the random sets were compared. So 6 were found to be better in with respect to this coverage. However, when the Gibbs free energy, which is required to obtain such a canonical set, was compared, the canonical set was found to be better on the in total. So we see that in sum, this canonical set is optimized on many parameters, not only on one. And this is highly interesting and a very strong design argument. So, and this is what the group, how the group commented on this result. Sets that cover chemistry space better than the genetically encoded alphabet are extremely rare and energetically costly. So we see that in total the canonical set is the best one. 4 years later, in the last publication, they stated that there is mounting evidence that the modern genetically decoded amino acid alphabet is highly optimized for a number of features. So the finding here is, the conclusion here is, the canonical set of amino acids is highly optimal. Nevertheless, the group arguments that somehow evolution made it. They write, it seems reasonable that several mechanisms led to the evolutionary selection of the canonical amino acids as they were made available during the emergence of metabolism. How exactly these mechanisms should have worked, they do not really explain. They only say that it is somehow reasonable. Now, how about nucleotides, the other kind of monomer, which is important for our genetic material, the nucleic acids? Nucleotides are quite rather sophisticated molecules when compared with amino acids. I have manipulated them in lab quite often, and I can say that you wouldn't obtain them if you don't use a multi-step procedure, if you don't use protective groups, if you don't use inert gas, and so on. It is difficult to obtain such building blocks. And also, it is not easy to manipulate them because they are rather unstable. For me, it was striking that such molecules are at the base of life. I couldn't imagine that such molecules can form somehow by unguided chemical reactions. And even more so, these molecules are compatible. They are size compatible, so that they allow the entirely reversible association and dissociation of nucleic acids. There were many alternative building blocks synthesized. I did so also in laboratory, and when nucleic acids are manipulated chemically, then often the chemical properties change in a way that is unfavorable. Now, let's come to the macromolecules. I'd like to start with proteins. Proteins are obtained when amino acids are joined, forming peptide bonds. And this polymerization reaction is demanding because chemical activation of the carboxylate has to be performed. And the carboxylates are among the most inert groups in organic chemistry. Also, water has to be removed from reaction mixture in order to shift the equilibrium to the right. Otherwise, this reaction won't work. So in a prebiotic setting, this would be rather improbable because water, of course, is abundant. And once a peptide chain is obtained, it can fold. It can either fold locally to alpha helixes or beta sheets. This is the secondary structure. And it is quite straightforward to predict a secondary structure from a primary structure. And this is also interesting because one can say that these molecules are really ingenious macromolecules because you can predict the secondary structure from primary structure. But until now, it is unclear which principles exactly are behind the formation of tertiary and quaternary structures. At least it is difficult to predict them. But well, it is reasonable to assume that the primary structure strongly influences also the tertiary and the quaternary structure. And proteins can be subdivided into two groups. There are the ordered proteins, which are mostly, or which are quite often enzymes. And life has to solve one main problem. This is the performing endergonic reactions, because most metabolic reactions are endergonic, and this is why ATP is necessary. And this requires proteins that can perform energy coupling, meaning that ATP is hydrolyzed in one subunit and the endergonic reaction here is performed in the other subunit. And there has to be an energy transfer. So this resembles a kind of a motor. And we all know that constructing motors is a difficult job. This is also why Frank Harold, a biochemist, wrote in his book In Search of Cell History, to the best of my knowledge, no one has yet described a single reaction that couples plausible prebiotic energy source to the production of organic compounds. So this is a very difficult problem to solve, and it doesn't look like it will be solved at all for prebiotic chemistry. The other group of proteins are intrinsically disordered proteins, and it's not long ago that they have been discovered. Until the 90s, things were rather simple about proteins. It was held that proteins usually form a stable fold, meaning that they are that they form one fold and that they have one function. But today, according to calculations, 30% of the proteins in humans are intrinsically disordered proteins. And this means that they have no stable fold. They can adopt many different folds. And they have many functions. And multifunctionality is something that any engineer would like to realize in his devices. So, and intrinsically disordered proteins are fascinating because they can interact with various substrates that share no structural homology. And this was unexpected and caused a lot of astonishment among the experts. Dyson and Wright commented on this in one of their publications. Indeed, it has been pointed out that in the presence of the coupled folding and binding of intrinsically unstructured protein domains, the proteins would have to be 2 to 3 times larger in order for them to form such extensive interfaces, which would either increase cellular crowding or the size of the cell itself. So we see here that this is again a design indication because these proteins are highly optimized. They are used in a very efficient manner. Of course, the question here is, can unguided processes generate high information or function density. And for nucleic acids, this is similar. Nucleic acids are formed from the condensation of nucleotides. Again, here the chemical activation of phosphates is problematic because phosphates are also among the most inert groups in organic chemistry. An organic chemist would always try to avoid using phosphates because phosphates are hard to introduce into a molecule and they are hard to remove. And also, phosphates are hard to activate. And condensing such molecules is really tough. And at the same time, in this reaction, also water has to be removed, similarly to the formation of proteins. Otherwise, the equilibrium will be shifted to the left, and there won't be any formation of nucleic acids. So once nucleotides are joined, we will obtain poly-anions. And this is interesting because one would expect a poly-anion not to join with another poly-anion because there is electrostatic repulsion. And Stephen Banner comments on this in one of his publications. In the design of a molecular recognition system, charge complementarity might be sought. Few chemists would design a poly-anion to bind to another poly-anion. I will explain later on why this is important. So physical properties of these polymers are independent of their sequence. That means the solubility and the folding propensity of nucleic acids won't change if the sequence is altered. This is totally different for proteins. In proteins, a single point mutation can change the whole folding. And according to recent findings, well, the evidence is growing that there are no nucleic acids without a purpose in organisms. Recently, there was a publication where the term pseudogenes was questioned or even said to be misleading. Cheatham et al. Write, where pseudogenes have been studied directly, they are often found to have quantifiable biological roles. Now let's compare proteins and nucleic acids. Nucleic acids are genetic molecules, and they have the following general properties. They are composed of few building blocks. They have a weak tendency to fold, and their properties are independent of sequence, of their sequence. For catalytic molecules, this is exactly the opposite. Proteins are composed of many building blocks, which is required for variability of phenotypes and also for the specificity of the catalysts. They have a strong propensity to fold in order to form cavities, which is also necessary for catalysts, and their properties depend on their sequence, which is also indispensable for a catalyst to be specific. And this also is interesting, as commented by Stephen Banner. The terrestrial version of true biopolymer life contains a paradox, however, relating to its origins. It is difficult enough to envision a non-biological mechanism that would allow either proteins or nucleic acids to emerge spontaneously from non-living precursors. But it seems highly improbable that both biopolymers arose simultaneously and spontaneously, and even more improbable that both arose spontaneously, simultaneously, and as an encoder-encoded pair. So to summarize this, it seems to be necessary for life to start from genetic molecules and catalytic molecules. But theories on the origin of life usually start from a one polymer system. Now, I'd like to remind us that replication and genetic code are also quite sophisticated and highly optimized. I won't go into detail on replication. I think most of us know this process to be very complex. It requires 20 amino acids, 20 specific transfer RNA molecules, 20 amino acid tRNA synthetases, which charge the transfer RNAs with amino acids, and a highly sophisticated molecular machine called ribosome. And in nature, there is no simpler version. And for the genetic code, it is, as already stated by Zachary Arden yesterday, it is highly optimized. It is well known that one amino acid is coded by 3 nucleotides. And already in the 90s, it was found that the genetic code is highly optimized. Freeland and Hurst gave their publication the title, "The genetic code is 1 in a million," which means that it is the best one of a million, or it is better than a million of other genetic codes that were obtained by random processes. And other interesting findings have been found out about the genetic code. For example, that it exhibits regular redundancy. It has a remarkable error robustness. It has a non-random distribution of amino acids among the codes. That means that the assignment of amino acids to the codons is highly non-random with respect to polarity and bulkiness of amino acids, the biosynthetic pathways of amino acids, the reactivity of amino acids, and even the taste. And also it was found, and this is a paper that I highly recommend, it was found that it contains an intelligent signal which can be extracted by means of straightforward mathematical operations. So we have seen that, and this is a very strong design indication. And the question is, how about the last universal common ancestor? Which are the minimal conditions, or which are the minimal prerequisites to make a cell viable? One can also call it a hypothetic minimal organism. So it requires the following genetic modules: a module for the synthesis of genetic material, for glycolysis, synthesis of the cell wall, synthesis of cofactors, transcription, translation, replication, and fatty acid synthesis. In sum, these are 250 to 300 genes which are required. And of course, don't forget a system of excretion and selective uptake. What has not been mentioned here is the module for cell division. There is the speculation that it is not necessary for a cell to grow. However, this is questionable. And often Luca is assumed to have been an anaerobe. And this requires a closer look to anaerobes. However, anaerobes are not really well understood. One of the organisms which are best investigated is Clostridium cluveri. It ferments ethanol and acetate to butyrate and caprate. And also it produces molecular hydrogen. And producing molecular hydrogen at atmospheric pressure is energetically costly. This is why, in general, this reaction seems to be not really efficient. It yields roughly 1 ATP molecule energetically. And 15 or 20 years ago, it was not understood how actually such organisms make a living. And Buchel and Tower wrote in a review, because of this result and work by others, it was assumed that anaerobes were inefficient energy converters, because they were only able to conduct structure-level phosphorylation, but not electron transfer phosphorylation. So I think this assumption was also made because people had evolution in their mind and assumed that anaerobes, of course, are more primitive than other organisms, and it is reasonable to assume that they are inefficient energy converters. However, during the last 10 years, the opposite was found. Anaerobes contain proteins that are highly efficient with respect to electron transport. These are called electron bifurcation proteins. What happens there? One cofactor transfers an electron to one side. This is an exergonic reaction. This is an exergonic reaction. And this is an endergonic transfer of the electron. And both energies are quite similar. So again, here is a compensatory process which minimizes the loss or the dissipation of energy in a protein. And this is something we find in anaerobes, which are assumed to be the most primitive organisms. So Zhang comments on this with the words, although electron bifurcation is carried out with near perfection from the standpoint of energy conservation and electron delivery yields, it is a biological energy transduction paradigm that has only come into focus recently. Remarkably, bifurcating electron transfer, proteins typically send one electron uphill and one electron downhill by similar energies, such that the overall reaction is spontaneous but not profligate. So this is highly interesting that in anaerobes such proteins were found, which were assumed only to be present in eukaryotes, because they are, of course, more sophisticated. They are assumed to be more sophisticated and more highly developed organisms than anaerobes. Now, which approaches are there for the explanation of the origin of the first cell? And there are two kinds of approaches, the RNA world and the metabolism first scenario. In the RNA world, nucleotides are obtained by unguided chemical reactions from small organic molecules. Later on, these nucleotides are supposed to polymerize to obtain nucleic acids and also to replicate. Then proteins join this ensemble to contribute catalytic efficiency. And finally, this whole thing is wrapped up by a membrane. In the metabolism first scenario, the small molecules first organize to a cycle. Then nucleic acids stabilize the cycle by saving the information which is necessary for the proteins which drive this cycle. And a lipid membrane also creates a cell where this whole thing is enveloped. Now, which arguments are put forth for the RNA world? RNA acts both as, or RNA was found to act both as a genetic tape and as a catalyst. RNA is less complex than the ensemble of DNA, RNA, and proteins. So here again we have the intention to start with a one polymer system. Of course, this is simpler than to assume that two polymers originated immediately. And ribozymes are assumed to be molecular fossils. And the catalytic properties were found in the splicing reaction, where RNA is able to cut itself. And during the last 50 years, this theory was accepted by many scientists, so it is quite popular. As Higgs and Lehmann wrote, in the last 50 years, this idea has gone from speculation to a prevailing idea. However, the question is how the nucleotides might have been formed. How was polymerization proceeding? And also, what about the replication? Is all of these, are these processes possible? And for the metabolism first scenario, the following arguments are stated. Of course, it is less demanding with respect to synthesis than in RNA world. And also, catalytic cycles are at the base of life. And especially autocatalytic cycles are important because their molecular species reproduces itself. Here we have this catalyst which reproduces itself. So it is an autocatalytic cycle. In nature, there are some examples for autocatalytic cycles, such as the Kelvin-Dark cycle or the reverse citric acid cycle. Catalytic cycles are, for example, the citric acid cycle or the urea cycle. Now, here it is difficult to explain, or the question is it possible that specific catalysts might have been present at this first stage? Is it plausible that aggregation of proteins was somehow inhibited? Because this is a problem which occurs quite often in protein synthesis, even in lab. And how about closing cycles? A reaction chain will not necessarily close the cycle, meaning that the last product which is formed will not necessarily lead to the production of the initial molecule. So, and for the origin of LUCA, the following scenario is proposed. First, there is the pool of organic compounds, such as nucleic acids and so on. And then such progenode communities are formed. Progenode communities are, so to say, cells which are in between the LUCA, a real cell, and purely molecules which are not organized. Now, here again is the question, if such progenode communities are viable at all, how do they do autopoiesis? How do they maintain themselves? And how do progenodes propagate? Or how about the first cell division of such protocells? And I would like to briefly show the problems of the RNA world concept. One of the most cited articles is the one of Matthew Pauner, which was published in 2009, where starting from cyanamide, he and his colleagues obtained cytidine monophosphate. However, they have to add these compounds, glycolaldehyde and glyceraldehyde, and also diphosphate. They have to add it in a special order. So this order cannot be changed. Also, they isolated intermediate products, and they use concentrations, for example, of the buffer of 1 molar, which is not really prebiotic. And Banner comments on this experiment with the word, in Shapiro's view, the need for temporarily separated supplies of glycolaldehyde and glyceraldehyde is more than a problem beyond of the scope of this work. It is a fatal flaw. How about the polymerization of nucleotides? There, it is often made reference to the works of the Ferris group. They did experiments with activated nucleotides, which are depicted here. This is such an activated nucleotide, and they used mineral clays also, and they obtained chains of roughly 50 nucleotides in length, which were physisorbed at the surface. However, the main problem again is that such activated nucleotides are highly unstable. They had to change, during the experiment, they had to change the solution daily in order to make the reaction proceed. Otherwise, the length of the polymers would not be closely as long. So they stated themselves a problem with the RNA world scenario is that the absence of a plausible prebiotic synthesis of the requisite activated mononucleotides. So how about the replication? So replication is when a polymer is obtained, and then after that, activated nucleotides anneal to this polymer. And afterwards, they form chemical bonds, and this duplex has to dissociate. This dissociation happens, of course, at elevated temperatures. And the renaturation, meaning the annealing, has to happen at lower temperatures. I have performed several experiments, or many experiments, on the denaturation of DNA, and I have to say that this expectation is not realistic, because even nucleic acids with a length of 20 nucleotides dissociate at temperatures above 60 degrees. So if such nucleic acids are longer, they will be hardly separated even at 90 degrees. And at elevated temperatures, RNA degrades quite quickly. So another problem is that this replication, this enzyme-free replication, is very slow. The best value is roughly 1 monomer per day, and this is really the best value. In some experiments, it was even 10 days per monomer. That means 1 monomer is added to the growing chain per day. This is very slow. Also, these reactions are highly error-prone and always incomplete. And of course, finally, RNAs are generally unstable to hydrolysis, and there are many bonds that are unstable to hydrolysis. The phosphate, the phosphate bond between nucleotides, and this this bond here, and also the base itself. So they are all prone to hydrolysis. This is all known, but nevertheless, many chemists, many scientists somehow believe RNA might have been formed. So let me briefly show some aspects about the metabolism first theory. It requires the synthesis of amino acids, and there are many Miller experiments have been performed, but the main product of Miller experiments are carboxylic acids. And in many such experiments, significant amounts of glycine and alanine are obtained, but all the other amino acids are only obtained in small amounts. And there are several amino acids, especially complex amino acids, such as tryptophan, phenylalanine, tyrosine, and arginine, which are obtained rarely or never. So actually, Miller experiments show the opposite of the argument which is often put forth in media. They show that under these conditions, the canonical amino acids set will not be formed. And for the polymerization of amino acids, this is even more difficult, because when amino acid sequences become longer, they might be strongly hydrophobic and they aggregate. I have performed quite often peptide synthesis in lab, and some sequences, even though they are quite short, are inaccessible synthetically because they are hydrophobic and they aggregate. They have obtained rather modest or even bad yields for tripeptides, and the concentration of the gaseous molecule which triggers the polymerization was incredibly high, with 400 millimolar. So, and also when we imagine such a cycle, to start off, there have to be enzymes which are specific. And for example, this is the reverse citric acid cycle. And many of those compounds are structurally highly similar. And when the enzymes are not sophisticated and specific, they will not be able to discriminate between them. And they will cause disruptions of such cycles. So this is a problem which has been found by Orgel. He says, one needs, therefore, to postulate highly specific catalysts for reactions. It is likely that such catalysts could be constructed by a skilled synthetic chemist, but questionable that they could be found among naturally occurring minerals or prebiotic organic molecules. And finally, aggregation of proteins. This is what I have already mentioned. When in modern cells, proteins are kept from aggregating because there are chaperones. And whenever protein is misfolded, the chaperone repairs it. And if chaperones are not present, then what will prevent aggregation to happen in primitive cells? So this is, again, another problem which is significant for the metabolism first scenario. So on the formation of and the propagation of protocells, there are only so far speculations. I will just give briefly some citations from a recent publication on this topic. Due to the aforementioned water problem, should any catalytic polymer, let alone one so complex as a primitive ATP synthase, be formed by chance in a vent environment, without the constant repair and resynthesis by the enzymes of biology, it would soon be disassembled by hydrolytic decomposition. Progenode communities fail until selection discovers some form of photoautotrophy, supporting a much more active energy-coupled metabolism. And they also comment on the possibility of the first cell division. This is interesting to see how they comment on this. The final transition to life could have been facilitated through the chance combination of a fortuitous division of a compartment occurring just after the duplication of two sets of membrane-bound polymers, each pushed into opposite ends of an elongated protocell. So there is a lot of fortuitous chance combination, but there is no explanation how this should actually really happen. There's no mechanism which is explained in the whole paper. Yeah, let me conclude on and finish my talk with two sides of Frank Harold. Yet we seem to be little closer to enlightenment than were Oparin and Haldane, who pioneered this line of inquiry 70 years ago. And he also states that actually there is no reason to believe in evolution. But nevertheless, he does so. He writes, there is also no observational evidence for any inherent drive towards greater complexity and sophistication. Nevertheless, most biologists, including myself, are satisfied that standard mechanisms suffice to drive life up Mount Improbable. So, and my personal conclusion is we have no findings that support the possibility of the origin of life through chemical evolution. On the other hand, there is a plethora of findings which clearly speak against this possibility. Based on the present state of knowledge, the origin of life is impossible without the intervention of a creator. Thank you very much.